welcome everyone. Welcome back. Today we'll have a treat of, we're here in this colloquium where we've been at this intersection between machine learning and biomedical sciences, really going both ways. We'll actually be talking more about applied mathematics and biomedical sciences and how to think about foundations in applied mathematics in particular and topology, and then really motivated by, particular biological questions and really going both directions, so from biology to foundations and then from the foundations back to biological questions. And so I'm very excited to have Kathryn Hess here from EPFL. So she got her PhD in algebraic topology actually here at MIT. And then held positions at the University of Stockholm, Nice, and Toronto before going to EPFL, where she's now professor of mathematics and life sciences. So her research focuses on algebraic topology and its many applications, primarily in the life sciences, a lot of neuroscience applications, but then also some applications in material sciences. And she has, for example, developed a lot of topological data analysis methods that were used for high-throughput screening of nanoporous crystalline materials, as well as, and I think we'll hear probably a lot of that today, the classification of neuron morphologies and also neural network dynamics. Kathryn has been highly recognized for her work. She was elected to the Swiss Academy of Engineering Sciences, was named fellow in the American Mathematical Society, as well as the Association for Women in Mathematics, et cetera, et cetera. So I'm very excited that Kathryn is here. She's enjoying her sabbatical and came to visit us during that time. So I'm very excited to have you here. So the floor is all yours.
Thank you very much. Thank you very much, Caroline. Thank you for the invitation. Thank you to everyone for being here. It's a great pleasure to be here. I've really enjoyed the time that I've spent at the Schmidt Center so far. And I really look forward to the coming days as well. Please feel free to make appointments, stop by. I'd love to talk with people. That's what I'm here for. Recharging my research batteries after 4 years of being an administrator. So I'm really eager to hear about your ideas, what research you're doing, and any ideas that my research inspires in you. So today I'm going to talk about a project that was led by one of my former PhD students, Kelly Maggs, and one of my current PhD students, Marcus Youssef, together with some people who are from the life sciences at EPFL or life sciences at the University of Lausanne, and now Heather Harrington, for whom Kelly is a postdoc in Dresden at MPI. And this is research that goes back to the roots of Kelly's thesis project. So he was hired on an international training grant from the European Union, which was all about cancer biology and innovative methods of doing cancer biology. And his specific project was to think about how the menstrual cycle affects gene expression. And so we're really looking for topological tools for understanding the cyclic behavior in gene expression. And it really took until basically the last year of his thesis to have the right mathematical tools in place. And so this, what I'm going to be talking to you about today, is really this project. What kind of mathematics goes into understanding cyclic behavior of gene expression? So what kind of challenges are we talking about? So when I first started thinking about this kind of data, it was before there were even single cell. We were thinking just about ordinary RNA-seq. And now I'm going to be talking about what sort of problems can we tackle with related to single cell data for which we can use tools from topology and geometry. So the first thing I want to talk about is identifying cyclic processes. So we're interested in processes that are going back to homeostasis, for example. And so what are the steps that go into our method for identifying such cyclic processes? So we're going to use tools from topology, specifically something called persistent cohomology, which is a tool from topological data analysis. And the pipeline, we can develop a pipeline to detect and compute the statistical significance, of circular structure in such single cell data. So we not just detect it, but say, okay, what is it really significant, what we're detecting there, and how can we justify it? So the tools we'll be using are from topological data analysis. So I'm going to explain these pictures here, but basically we're going to have a signature of the data that looks something like this, which is called a persistence diagram. And you see that this point here is pretty far from the diagonal, and that turns out to give us some notion of statistically significant cyclic processes. And I'll explain in more detail how that works. The other type of problem we want to study is, okay, once we've identified these cyclic processes and we can say sort of where in the process the different cells are, we'd like to see something about which genes are actually involved in these processes and in which order are these genes activated through gene cascades. So what we're going to do is now use geometry, another kind of mathematics, to really analyze the dynamics of the gene activity in these cyclic processes. And we're going to look at this by looking at the embedding of this circular structure in transcriptomic space. And so what we'll end up with in the end is a pipeline for estimating both cell and gene phases from circular structure. So we're going to have some sort of matrix that's going to tell us about which genes are leading which other genes. And from that, we'll be able to sort of see the different phases and amplitudes of the genes as we go through the cyclic process, be able to say, oh, gene I is leading gene J, which is leading gene K. So that's the basic idea. And I'll explain in detail how we actually get there. I'll provide some validation type results on both synthetic data and well-known cell cycle data, and then look at some experiments we've done with different kinds of androgen receptor silencing. So the philosophy that we have is that if you look at the epigenetic landscape, this is Waddington's famous epigenetic landscape, that the topology and geometry of this landscape, which is some sort of geometric object, actually has biological meaning. I don't think that's very controversial. But how do we go about measuring the topology and geometry? Well, if we think about paths in this landscape, we can think of these as really corresponding to developmental trajectories, right? As a cell evolves, its gene expression evolves, and so you can really think of that as some sort of developmental trajectory. Now, in particular, if we have a cell that starts looping around in this landscape, then that's really some sort of cyclic process that we're seeing. Now, what turns out is that topological features of spaces, so topology is about the essential shape of a space, not having the sort of scale invariant, continuous deformation invariant, and that gives us a sort of stability to these features that we detect, and that provides us with robustness for the signature we're detecting in this way. And then we're going to think of single-cell data as really providing us with a finite sample of this landscape. We sort of figure out where the cells are moving in this landscape by looking at some sort of finite sample. Okay, so let's focus for the moment on circular enrichment. We're going to talk about the methods here, and then we'll see how to apply them later. So suppose we have some single-cell data. What do we do with it? Well, the first thing we're going to do is to find some sort of hallmark gene set. Say, in the context of the particular problem I'm considering, which are likely to be the genes that are important? Because it's very hard to analyze genes in a 20,000 dimensional space. We want to restrict to something where we have a greater chance of actually being able to analyze the data in a reasonable way. And so we say, okay, if we're thinking about a particular kind of problem, then we should restrict to maybe these 100 genes or these 200 genes or something. That makes it easier for us to detect the circular structure we're looking for and gives us some interpretability in the bargain. From there, we're going to simplify even further. We're going to project onto the first k PCA coordinates. Often in the calculations we've done, k equals 5 turned out to be a good answer. That's more of a not something that's mathematically justified, but it's worked so far. It's an engineering type solution. And then from that, we'll compute what's called the one-dimensional persistent cohomology of this point cloud. So what we're getting is a bunch of points in some sort of usually 5-dimensional space. We like to say, what kind of structure are we detecting there? And we're going to use the tools of persistent cohomology to detect these circular structures, and I'll explain how. And then we have a way of associating of detecting whether or not the circular structures that we've noticed are actually relevant, actually statistically significant, by associating to them some sort of score, which is called a ring score. And then we can evaluate the statistical significance of these ring scores, which will tell us something about whether the circular structure we've detected actually is meaningful and worth evaluating from a biological point of view. So how do we get from point clouds to what we need to use, which are called persistent diagrams? So we'll do an example in more detail in a moment, but the basic idea is if we have a collection of points in some low-dimensional space, if it's just a collection of finite points, there's no particular topology or geometry that comes out. But we'd like to say, well, if we have these collections of points here, it looks like we have two circular structures. Is there some rigorous way we can say that? So using the tools of persistent cohomology or persistent homology from topological data analysis, it gives us a way of making rigorous the argument that we have two circular structures here. This idea of taking little balls around each one of the points and slowly growing the radii and seeing what sort of more complex geometric structures arise. We see that once the balls are big enough, that we have two holes in the center here, which is giving us circular structures. And if it grows even more, then it all fills up. So what we can do as the radii of the balls increases is we can say, OK, at what point does such a structure arise? And at what point does it disappear again? So on the one hand, we can compute what's called the zeroth homology, which is just counting the connected components. Or we can look at the appearance and disappearance of these sort of circular structures, which is what we call the first homology. And then you can graph this here by saying at what time, at what radius rather, is this structure born, the radius of these little balls is this structure born, and at what radius do these things die? Does it all fill up? And so we could plot points, time of birth, time of death, like this. And looking at the distance from these points to the diagonal here gives us what's called the persistence of the feature. So this particular feature here corresponds to a persistence of 1. This feature here gives us a slightly shorter persistence. So what we look at is the difference between the death time and the birth time. And what we're going to do is, as in this image here, we're going to order the death and the birth time from longest to shortest, like this. So we're going to go from a collection of points by this method to now a sequence of non-negative numbers like this. And from this sequence of numbers, we'll be able to tell us something about the significance of the features that we've detected there. OK. So let's look at an example. So here we have some collection of points in the plane, like this. And I'm going to slowly grow these little balls that are centered around each of these points. So here we're supposing the balls are of radius 0.3. We grow them a little more, 0.5, and we start to see different structures emerging. There's a little circular structure. The idea is that you're going to add an edge between 2 points anytime the 2 little balls intersect. We grow the balls a little bit more. We see this circular structure still here. We see a hint of another circular structure that seems to be on its way to being developed. And we get to 0.9, and all of a sudden we see this nice big circle here. This looks like some sort of more significant circular structure that's showing up. And we'll say, OK, this is some sort of critical threshold radius, critical radius for these around these balls, for these little balls, at which we see something that seems to be a really important feature that's coming up. What we want to do is study this collection of points and edges that we get this way. That we can study using the tools of algebraic or combinatorial topology. If we continue growing these balls like this, then it starts filling up more and more until at some point, it's all filled up. And there's no more really interesting topology in the picture. So what are we going to do in this case? We're going to go back to this critical radius of 0.9 as we had. And we're going to find a way of encoding this circular structure by what's called a one cochain. So if we assign a real number value to each of these edges here, we can assign a real number value in a way that gives us just a function from the set of edges of this structure. So we're looking here at the collection of points and edges coming from the intersections of these little balls. And we wanted to define a function from the set of edges here to the real numbers. And so this is actually this set of functions from the set of functions from the set of edges to the real numbers actually is itself a real vector space. And we can make a specific choice of an element there that encodes this circular structure. There are actually infinitely many equivalent ways of choosing this encoding. And in the end, I will provide you with a way of making a specific good choice of a way of encoding this structure by a particular choice of a function from the edges to the real numbers. And so this vector space here we call the vector space of one cochains. That's just the mathematical language we use on this set of edges. So this is the standard way of encoding this kind of circular structure that shows up. And so now if we go back to our persistent diagram, where recall that we looked at the actual persistence, we look at the difference between the death and the birth, and we've ordered them from largest to smallest, then what we can do is associate to a sequence like that of persistences a certain score where we take this first persistence, this longest one, and we subtract from it the sum of the other persistences, where we divide the P1 by 2, P2 by 4, and so on, because somehow the shorter the persistence, the less significant it should be. It's somehow saying, how important is this feature that's picked up by this 0th persistence compared to the other ones? And that's what we'll call the ring score, where here we've chosen this n just so that the values of this score are always bounded between 0 and 1. And the idea being that the greater this score is, the more significant the circular feature that's detected by P0 is. And one nice thing about defining it this way is that this ring score is scale invariant. So if you have your collection of points and you apply a change of scale so that everything gets expanded like this, the significance of your fixed feature is not going to change. You could also choose, instead of looking at death minus birth, you could take di divided by bi. That's another, looking at what's called persistent ratio is another interesting thing to look at. But here we'll consider just these differences here. So using these ring scores can tell us how significant the circular feature that we've detected is. So how are we going to determine its statistical significance? So let's go back to the case that we're really interested in, which has to do with expression data for different genes. So suppose we've decided we're going to focus on M genes, and we have some point cloud of expression data in this M-dimensional space, for which we've computed this persistent cohomology, we've detected a circular feature, we've computed its ring score. You say, okay, the ring score seems big, but is it really statistically significant? And so what you could do is a permutation test to come up with an empirical p-value to tell you how significant this feature actually is. So what we did was, we have this huge set of 20,000 genes. We can take 2 to the 10 possible sets of M genes and do the same analysis on these randomly chosen sets of genes, which then gives you a null distribution of ring scores. Because it could be that if you just make a random choice of genes, you're also going to be finding these circular structures. But how significant, so this gives you a way of deciding, when I've made this specific choice of genes using my hallmark gene set and whatever, am I really getting something significant? So then you can calculate, once you have your null distribution, an empirical p-value for your original ring score and decide, okay, is this significant or not? So that's what we do in order to detect the circular structure and be able to say something about how significant it is. So there we're looking in this space where for each cell, to each cell we have a point in this space, the coordinates of which correspond to genes. Now what we want to think about is, okay, once we've detected this structure, what actually is the process? What cascade of genes is leading to this cyclical process? So remember that when we have one of these statistically significant circular features, then it's going to be encoded by one of these one cochains, one of these functions from the edges of the complex to the real numbers. So what we want to do now is to be able to assign phases to each of the individual cells once we've made a choice of what's called a harmonic circular coordinate. And that's what I'm going to explain to you, how once we've made a choice of this encoding alpha, we're going to make a good choice, so in some sense a canonical good choice of a circular coordinate. And then, once we've done that, we're going to be able to now look at the gene expression and figure out which gene is leading which gene in the process and what the phase difference is between them and the amplitude and so on. And for that, we're going to need more geometry. But to do either one of these stages, we need to introduce some geometry. So what is the story of harmonic representatives? So just to mention that the original work on such things went back to work of De Silva, Pike and Park, and Pereira. So what I'm describing here is a way of sort of integrating and amplifying a bit the work that they did. So recall that we had, at some particular threshold in this process, in our case it was 0.9, we had this statistically significant circular feature that appears. So we're there. right? And so R is this critical threshold at which this structure appears, all right? So now, what I'm going to do to introduce geometry is to introduce inner products, because once we have inner products, I can talk about angles, I can talk about distances, I can talk about norms, and things like that. So what I can do is then choose inner products on the spaces of functions from the set of nodes to the real numbers, and on the set of edges to the real numbers. So these are these one cochains we already talked about, and these other ones are called zero cochains. And there's a way of cleverly choosing this inner product that corrects for the fact that this cloud of points has non-uniform density. Because if your density of the cloud is very unequal, then if you're a little bit naive about how you choose your inner product, it's not really taking into account the geometry, the natural geometry of your space of points. So there's a way to do this that takes this point cloud density into account. And then now you can define the inner products on these real vector spaces. And once you have these inner products, we're in the world of geometry. So once you have inner products, then there's a way of computing a discrete Laplacian on these vector spaces. And when you have a discrete Laplacian, then you can start taking harmonics, which are kernels of the Laplacian. So you can find a unique representative in here, a unique one cocycle. So map from the edges of this collection of points and edges to the real numbers that really is going to encode your circular structure, and then you can extend in a certain way to a map to the circle. And the way to think about this is it really has some map to the circle that has the smoothest gradient with respect to the inner products you've chosen. So I'm not going to go into details about how you do this, but just to say it is possible to choose a circular coordinate like this. And then what you want to do, once you have that, you can determine the phases of the nodes of this complex VR, which correspond to your cells, and can tell you where in the circular process each one of these cells actually was when the single cell assay was done. All right, so how does this work? So we're back in this. Here's our critical threshold, 0.9, where we had this circular structure that appeared. And so now we have our circular coordinate, this map theta h, so it's corresponding to our choice of harmonic representative. It gives us a map from here to here. And this idea of determining the phase of the cells is using this map here, so that we can now use this to assign phases to the points via this coordinate. So if I have a blue coordinate here, I look at which points were over that, green there, and so on. So it's a way of assigning phases to the points in the point cloud. Quite straightforward once you have this theta h. But we can do more. That's just still in the world of what's going on with the cells. We want to say what's going on with the genes. How can we figure out which gene is leading which gene to figure out the gene cascades? Well, before I do that, just want to mention what's going on, the difference between theta h and alpha h. So theta h was giving us a way of assigning a circular coordinate to the different points. So here we're in the space where each one of these is a cell, in the space where the coordinates are actually genes. And then if you think about what alpha h is doing, alpha h assigns a real number to every edge in this VR, right? And when you're assigning a real number to an edge, you can think of that like a vector field. It's assigning a weight to a path in some sense. So we have on the one hand a coordinate, on the other hand, you're also picking up this one-dimensional structure. And that's what's interesting. You've got both the zero and the one-dimensional structure here. OK. But on to figuring out which gene needs which gene. So there's something called lead-lag analysis, which was for which we're basing this on the work of Baryshnikov et al. Maybe they were the first ones to do it. So the idea is, suppose that we have here in this toy example, we have two functions, the blue function and the orange function. The blue function is sort of lagging behind the orange one, which is leading it. So it's the same function, but they're just shifted from each other. And if you plot for every x, if you plot the pairs of coordinates, as you go around, it translates into a curve like this in the plane. So you can go back and forth between these two representations of the same information. And so you're getting a closed curve from these two curves that are following each other. So you can think about this as giving you this pair of curves here is giving you some function from the circle S1 into, in this case, R2. And under nice conditions, which is usually what's going to happen with real data, you can actually extend it over the entire disk. So you think of the disk, the boundary of which is a circle. Then usually there's a way of extending this over the entire disk. Now, how can you use this interpretation in order to figure out, from this curve here, for example, which of these curves was leading the other, that the orange one was leading the blue one? How can we detect that? So here's our situation. We have some map from a circle into Rn, n equals 2 in this case, extended over the disk. So now we're going to look at this sort of angular momentum vector field in the plane determined by xi and xj. And we think about our curve as being embedded with respect to this vector field. And if you think of the integral around the boundary of the disk, which according to Stokes' theorem is just, you can also express in terms of the integral over the whole disk, then it turns out that the signed area of the region in the plane that is bounded by this curve actually measures the extent to which gamma I is leading gamma j. So I'm not going to go into the mathematical explanation, but just to say that this computation is helpful for finding out, basically, if it's positive, then I is leading j. If it's negative, j is leading I. Okay? So you can use this basic idea. So this is just about, closed curves in the plane. But now let's think about how we can apply this biologically. So now let's think about, again, a periodic curve. So it's a map from S1 into Rn. And suppose that each coordinate of this function, so gamma j is the jth coordinate, can be written as a product of some aj times, there's a fixed periodic function from S1 into the real numbers that is just, if I take some complex value sz on the circle, and I want to calculate gamma j of z, I have some amplitude that's changing, and I have some phase shift that's coming from bg. But otherwise, the function is the same. It's just the amplitude and the phase shift that are varying. So that's what you would call a chain of offsets in this case. And often when people think about a simple model, so a naive model of gene cascades, you can think about it as being this kind of chain of offsets. Now, usually a function like chi will have some sort of Fourier expansion that has many terms. If it has just one term, it's just a real part of of a first Fourier mode, then it turns out that this, if you calculate this lead lag that we had before, it's actually proportional to the sign of the difference between the phase shifts. And so if we take the whole matrix of these lead lag, then what it's encoding is precisely these phase offsets of the coordinates. So that's the interest of calculating this matrix. It's going to give you how these phase shifts are working with respect to each other. OK. So now we want to apply this in the case of our v at our critical threshold r, 0.9 in our case. So now we suppose we have our harmonic representative, our unique harmonic representative of this circular structure. So it's one of these one cochains. And what we can do is we're going to choose, if you're thinking about this complex given by these points and edges, it can be really complex. But one thing we know as mathematicians is that for a large enough n, you can find a way of taking this thing and embedding it, really embedding it, not in the computer science sense, but the real sense that it's injective. We can embed this affinely in Rn for large enough n. And then we're going to define a function for every pair of numbers I and j less than or equal to n from the edges of our VR, right, to the real numbers. That's defined as follows. It's basically an integral. So it's going to send an edge of this complex VR to the integral of this particular one form we were looking at before, this angular velocity type 1 form that you see again up here, along the path in Rn determined by the embedding of the edge. So if I have an edge in here, and I look at this embedding, it's going to embed it as some path in Rn. And I can integrate this angular momentum form along that path, and I get a real number. So I have this way of associating to every edge a real number. And that's exactly a one cochain, right? So it's giving me a one code chain in this way. And I'm using process. So I haven't yet used alpha H. All I've used is this embedding so far. How does alpha H come into the story? All right, so I have my alpha H, I have my affine embedding, and now I can define a sort of a generalized lead-lag matrix as follows. I'm going to say the ijth coordinate is given by taking the inner product of alpha H and this one cochain. Because remember, I defined an inner product on the one cochains, so on this vector space of maps from the edges into the real numbers. And so here I have a one cochain, which is alpha H. Here I have this one cochain that I just defined by taking the integral along these paths. And so it makes sense to calculate this inner product, and I get a real number. And this real number is again telling me something about who is leading whom. So if I think about what this is computing here, this is computing, for every I and j, it's computing the area of the parallelogram that's determined by the, so if I have an edge from a vertex to a vertex, and it's mapped by this affine embedding into Rn, each endpoint of the edge is mapped to a vector in Rn. If I look at the parallelogram that's determined by those two vectors, what this computes is the area of that parallelogram. And so this here is computing some sort of linear combination of these signed areas of parallelograms weighted by alpha H, which is taking the density into account as well. So it's some sort of very sophisticated version of what we saw with the two-dimensional thing. Now, this matrix here is skew-symmetric, which means that we have lovely ways of analyzing it, finding its complex eigenvalues, the eigenvectors, and so on. And you can look for the largest eigenvalue, or the ones that have largest real value. You can find the sort of principal eigenvectors. And if you look at the plane determined by the principal eigenvectors of this matrix, that's going to give you a nice plane, and looking at projections onto that plane can actually give you a way of inferring the ordering and the amplitude of the different genes. So that's a whole, there's a bit more math to do in that case. But the basic idea is that we have this really lovely skew-symmetric matrix, which we can analyze by standard methods from linear algebra, and come up with appropriate eigenvectors onto which we can project and determine interesting information about our genes. OK, so here's the basic idea. We're looking at these lead-lag profiles for different pairs of genes. It gives us our lead-lag matrix like this. And from that, we can determine the principal lead-lag plane like this, onto which we can project, and then determine from that what the components of the different genes are along this path. And it tells us how we can order them as we go around our cyclic process. OK? So that's the basic idea. So that's the math. Now, it seems like we've made a lot of assumptions along the way, assuming that we can use this sort of lead-lag analysis. And in particular, when I was saying, well, we can pick up the phase differences and so on, I was making this assumption that the dominant Fourier mode by far was the first. Real life is, of course, much more complex. Does this actually work when you apply it to real data? That's what we'll see next. So a bit of validation. OK, so first we're going to look at some synthetic data, right? So one always does first. So what kind of synthetic data are we going to use? Well, we're going to look at a linear ODE model of gene transcription and translation. And in particular, so I'll just click through here, we have a pair of ODEs where we're looking at the vectors of mRNA and protein levels for different genes. We have an interaction matrix that's describing the effect of the protein, of the linear effect of the protein on RNA production, et cetera. So the typical sort of ODE model that one would have for thinking about gene transcription. And then what we do is we make a choice of this interaction matrix that is going to force the existence of cyclic structure. Okay, so we know it's got to be there. And we say, can we detect it? Can we detect it properly? Are we going to get the right ordering of the genes and so on? Also, in order to make it a little more realistic, we're going to use with the negative binomial sampling that was proposed last year in Nature Methods. So it looks more like really noisy single-cell data. Okay, so it's not just going to actually lie beautifully on a circle. Okay. All right. So when we do this, we did it for a couple different examples. We look at one example of synthetic data where this was really the underlying interaction matrix between the different genes in terms of the strength of their interactions. And then this was the lead lag matrix that we deduced from the methods that I was just describing to you. So it matches quite nicely. Okay. And then what about, having the circular enrichment of the gene set that we were talking about? So by choosing PCA components appropriately, we ended up with very nice ring scores that were detecting actual circular structures like this. Okay, in the case where we're looking here at PC1, PC2. All right. And now if we do the cyclicity analysis with respect to the genes now. So this is looking at, here they're plotting the lead lag matrix PCA with its real value and its imaginary value, and we see a nice circular structure coming up, which then we can translate into the individual genes and see how they're plotted, what their particular phases are and their particular amplitudes, doing this kind of analysis that I was talking about before. That's just what it looks like, and say, okay, does it work? Are we picking up what we would expect to from the synthetic data? And so here we're plotting the gene index, which is really the order of gene activation according to the synthetic model, against the phase as detected by our method. And we see it matches quite nicely, correlation coefficient 0.986. And then you say, okay, well, what about the relation between the strength of the ground interactions and the lead lag that we detected? Not too bad, 642, the correlation coefficient. So pretty happy with how well we could extract the ground truth using this method. Another example with another interaction matrix, the corresponding lead lag matrix that we deduced by our method from this. And also here we see the correlation between the ground truth of the interaction and the lead lag was actually excellent in this case. And then you do, again, this cyclicity analysis. Again, we're looking at the genes now, where we're looking at the lead lag matrix with its real PCA and imaginary PCA here, and seeing, again, the distribution of amplitudes and phases of the different genes. And it corresponds very well to the synthetic model. Again, here we're looking at plotting the gene index, which is saying the order in which the genes are actually activated in this synthetic data. And then the corresponding phase, which is computed from the gene expression data. And we say that, again, it aligns very, very nicely with a coefficient of 0.998. And then we look at here, we also had there is a time element when you're looking at these ODEs. We actually have ground truth time and compare the ground truth time against the estimated circular coordinate. And since it's being plotted like this, you really see the circle here coming around and coming around like this. So that the circular structure maps very well with the time component of this linear ODE model. Okay, so that was nice validation with synthetic data. The other validation we did was with actual cell cycle data, which is something that's extremely well understood. And so the data that we took was from cycling human dermal fibroblasts from a paper by Capolupo. And the data set that we used was known markers of certain families of genes which are activated in the cell cycle. And so when we look at this, so here again, we're looking at the gene, the cyclicity of the genes. So here they're saying something about the counts of the different cells as you go around. And what you see here, the color scheme here is telling you about the activation of the genes. And you see that in this upper part of the cycle, you have the S genes that are being activated. In the lower part, you see the G2M. So this is, for the biologist, straightforward that this is the way it should be, and it is indeed exactly what shows up. When we apply this lead lag analysis to the actual data from Capolupo's paper. And again, you can plot where exactly the phase and the amplitude of these different genes are, the S and the G2M here. and the lead-lag matrix, which corresponds very well to what you see in real life with S activating G2M, and so on. So when we do this comparison with actual cell cycle data, you see that you see what you would expect to see. So we didn't learn anything from this except that we seem to be picking things up properly. And another thing that can be interesting to look at is here there are 5 different sample genes that we considered. And the color scheme again is saying where are things being activated. Here we have the phase moving in this direction. And for those who know what these different genes mean and when you would expect to see them activated, you can sort of see their activation moving around this cycle appropriately. Okay, so the reason I'm showing this is because it's just to illustrate the fact that this method enables you to say, okay, for a particular gene, when is it actually being activated? Where do I see? What is its phase, and so on? Okay, so where things really get interesting is not when you're just doing validation and making sure the model actually is, working appropriately, but it's when it can tell you something new. And that's what I'd like to tell you now is some new things. In fact, some of it is so new that I modified my slides after a conversation with Kelly this morning. So there's some stuff that he finished up over the weekend. So experiments. Right, so what I'm going to think about is a case where we're silencing the androgen receptors using viruses. And the first thing we wanted to do was to say, okay, well, at least in the control case, because we're going to do silencing on the one hand, and we're going to have a control case on the other. I'd like to see that at least in the control case that the cell cycle data looks like that of Capolupo, because we're looking at different batches from different labs, different pre-processing, different everything, but there's both a cell cycle. Do we see the same kind of results? So that's the first thing. And the second thing is, okay, what is the impact of androgen receptor silencing on gene expression? This is the sort of question you want to go and you perturb something, you say, what breaks, what doesn't break, and to what extent? And can we actually measure this and see this using these kinds of methods? So the data we had was single-cell RNA data-seq, again from human dermal fibroblasts. And we're using 2 different lentiviruses that were to express AR silencing and also a control. And then we're looking at 2 different hallmark gene sets, or maybe even 3. One was from VeloCycle, and the other was something called SASP that has to do with senescence. Okay, so what did we discover in this case? First of all, we looked at a comparison between just the cell cycle. And here we were looking at actually both the control and the AR silence genes, cells. And what you see is that when you compute the phases of the genes and their amplitudes, they correspond beautifully between the 2 different data sets. So one from the Capolupo paper I was talking about before, and then this new set done in the lab of Paolo Dotto. So a very nice correspondence there. It shows that this sort of topological or the geometric computation we've been doing actually is something that's stable across data sets, pre-processing, and so on. Okay. Right, so essentially identical phases and amplitudes, despite the different experimental protocols. Okay, now what about the existence of cyclic structures? So in the case here, we're looking at this SASP gene set. We do the persistence diagram. We see a point that is far from the diagonal. How significant is it? So it corresponds to do this permutation test. So its ring score is somewhere out here, which is very, very far from the rest of the distribution. So this would be your null distribution. And you have a very nice p-value. You say, OK, looks like I have a significant feature. And then you can say, OK, now we look at the gene cyclicity analysis. So here I would have the 2 different conditions, the orange being the control condition, the blue being the silenced condition. And what you see is that you get roughly a circular structure. It isn't as pretty as in the synthetic data, of course. But you do see basically a circular structure. And this is showing you for a couple of specific genes when they are actually activated. And one thing that is interesting, so far we haven't seen much of a difference between the genes in the AR silence case and the control case, is that here there's a way of measuring what's called the AR silencing score. So this is looking at the expression of the 200 most highly expressed genes in the case of AR silencing. And you see that there's a point in this cell cycle. So this is looking at sort of a three-dimensional version of what we had on the previous slide, where we have the lead lag PCA, real and imaginary here. And then we're looking at this AR silencing score. And you see there's a very specific point in the cell cycle where all of a sudden these genes are really expressed. And so one thought is, are we moving into some sort of senescence at this point, that you're sort of moving out of the cell cycle when you reach this point? Is that what we're seeing as a phenomenon here? So that's one hypothesis about how to interpret this image, that you have this moving away from the cell cycle here. And we'll see that this is confirmed in a certain sense. So what we'd like to see is how stressed the cells are when they're in this AR silenced mode. So what we're looking at here are looking at the entire data set, both controls and the androgen receptor silenced ones. And we're looking at the most highly variable genes and then projecting onto PC0, PC1. And what we see is that here we have, again, orange is the control and blue is the AR silence, that there are certain genes that are much, much more highly expressed in the blue than in the red. And we see examples here that we have these three genes, that are particularly highly expressed, coming from this light color here, in the cells that are AR silenced. So there's something very special going on with these genes in these androgen silenced cases. So if we look at that and say, okay, let's look at what's really going on with those genes, and we look at some measure of the stress as being how highly expressed these particular genes are in these AR silenced genes. And we have the following picture. So what this is showing us here, on the one hand, we're looking at the cell cycle coordinates. So the cell cycle is moving along here from left to right. And then on the y-axis here, I'm looking at the extent of this stress that's coming from the AR silencing. And so we have the different phases that are measured here. Everything that's in blue is the G1, orange is this G2M, and green is this S. And so that's just showing me where I am in the phase. But if we look now, again, at this division between the orange being the AR silenced ones and the blue being the control, we see that for the orange ones, there's a kind of a threshold. When the AR silencing stress goes too high, they just don't enter the cell cycle. There are some that can enter the cell cycle that are more resilient, where they have a stress level that's lower, but those that are above this threshold somehow get stuck. And so what we have is that the cells that are expressing high levels of this sort of androgen receptor stress don't enter the cell cycle, but those that are lower seem to be able to. So there's some sort of degree of resilience to what's going on. So it's sort of a phenotype that we're seeing expressed here in terms of how resilient they are to this androgen receptor stress. And then you can see that if you think about the ones that are this sort of AR resilient, the ones that do manage to enter the cell cycle, then they actually behave identically, essentially, to the non-AR silent cells in the cell cycle, because you have practically perfect correspondence between the gene phases and the gene amplitudes in these two cases. So there seem to be these really two different phenotypes for these human dermal fibroblasts. There are those that, when faced with this androgen silencing, they're just blocked, they're stressed too much, and there are others that manage to get over it and continue in the cell cycle. One observation is that the ones that do seem to get blocked seem to be ones that are maybe becoming more similar to epithelial cells. And we'll see why this may be relevant in just a moment. So specifically, now we're going to think about another case in which we have androgen receptor silencing. Well, androgen silencing, because we're going to think about a case where we actually are going to castrate some poor mice. And So all of a sudden, there's no more androgen, and see what happens. This is very different from using lentiviruses to silence androgen receptors, but what's going to the result the same. So what we're looking at is data from a study of prostatic epithelial cell regeneration from Carthouse in 2020, and we looked at 50 different hallmark gene sets. So the process is this: you castrate the mice, and then the prostate involves, and by 28 days it's reduced, I think, 90% or something like that. Then you add testosterone back, and it regenerates, and at the end of 28 days, it's practically back to normal. And then what the researchers did was to take samples at different times throughout this cycle to see what was happening with the cells. So you really have this time element, which is very explicit there. And so when you compare that to gene sets having to do with androgen response, or even having to do with cholesterol homeostasis, or even others, you do get significant circular structure. And you even get, quite nice p-values, even for myogenesis, which has to do with muscle rehabilitation. So we get significant circular structure with respect to multiple hallmark gene sets. And so you get the same kind of cyclic structure showing up for both the androgen receptor and the cholesterol homeostasis gene sets. And you can see here the time points, so the different colors here correspond to different days in this process, all the way from intact to the last day of castration, which is the purple, and then back to full halfway through the regeneration. So we have these nice circular structures. We can measure, again, the gene phase and amplitude. This gene, for example, is one that's highly known to be related to prostate, for example. And then you see a similar kind of thresholding effect coming in. So if you look at this plot where we're plotting the cell cycle versus tissue regeneration, as two processes that can be ongoing in the cell, you see that in the case of the intact ones, some of them are here and some of them are here. And you go along, and when you get to the 28th day of castration, just before they get more testosterone again, let's see, right there, you see that they're all concentrated here. They get blocked. They can't enter the cell cycle. We're seeing the same kind of phenomenon here with inability to enter the cell cycle. And there are very, very few that actually do enter the cell cycle. So there seems to be something about epithelial cells that makes it more difficult for them to, or maybe it has something else to do with the experimental protocol, I don't know. But in any case, in this case, they don't seem to be able to enter the cell cycle when they don't have androgen. So conclusions from this: first of all, using topology and geometry, we can detect these significant circular structures, which then enables us to determine phases of our individual cells. And then we can infer these processes underlying the circular structures. Some advantages: well, the first one is not going to be will be kind of controversial here. We actually don't use any machine learning techniques. But maybe the kind of output we're producing could be useful as input to different machine learning techniques. We've demonstrated that it's applicable over a wide range of experimental settings, different kinds of samples, different kinds of pre-processing, multiple gene sets, and so on. And we're getting robust across gene sets, across so on, 0 and 1-dimensional information. 1-dimensional information being information about processes. The 0-dimensional information being more about expression of individual cells, and so on. Limitations, because there are always limitations. There are hyperparameter choices that have to be made when we do the pre-processing, for example, which genes to study, and the impact of outliers. So this is something that's still a serious problem, can be a serious problem when one is applying tools from topological data analysis, although there are ways around this as well. Interesting future directions we'd like to consider. RNA velocity, of course, is something that people study a lot in the context of single-cell data. We have preliminary data showing if you look at the spliced versus unspliced for RNA, then you do see the sort of directionality you would expect from RNA velocity. I'd like to integrate the two together. I think that could be very powerful. One thing that would be interesting to study is really get more deeply into concurrent biological processes. Because one thing we've already seen, for example, when looking at comparing the androgen receptor cycle together with a cholesterol homeostasis cycle, you see these sort of things they're wound together in some sort of interesting, complex way. And also, how do you integrate together these sort of cyclic processes with more linear processes that are happening at the same time? And more experimental data. Definitely want to go there. So with that, I thank you very much for your attention and be very happy to answer any of your questions.
Thank you very, very much for a wonderful talk. Okay, questions. So maybe could you just go to the microphone and I will come here. Thanks.
Thank you so much. That was a great talk. I had 2 questions about the lead lag analysis. The first is, does it assume that the genes have very similar distributions? What if like the leading gene is expressed very differently than the lagging gene, I guess? And then the second question is, could you use it in a naive way where you don't previously define a gene set and you just kind of ask over the whole data set which genes might be leading others?
So those are both really good questions. I think that, yes, implicitly for the time being, we're assuming the distributions are very similar. Clearly, I mean, this is so far, it's, I would say, fairly naive. And it'd be really nice to make it a more careful analysis. I think it'd be a very good idea. That's a really good question. And as far as I know, we haven't thought about what happens if you don't have any prior knowledge of what genes are there. But I think it could be interesting to see whether there's a way of going about it in sort of, I mean, this would appeal actually to a topologist not to have to choose coordinates, right? To be able to pick out those sort of structures without having predetermined the coordinates. That's a really interesting idea. Thanks.
Hey, yeah, thanks for the great talk.
Thank you.
So yeah, precision cohomology is like a great technique for detecting circular structure. But the few times I've looked at like single cell RNA-seq data for like developmental trajectories, then rather than having a circular structure like you would have for like cell cycle or like menstrual cycle, like you mentioned at the beginning, you end up with like a kind of like branching structure. So from a topological perspective, that's a point.
Right.
But it seems like there's kind of similar methods that should be helpful for like still kind of like uncovering that branching structure. So I'm wondering if you have any thoughts on that.
No, that's a really good question, and I agree that, and this is one thing that Kelly is giving a lot of thought to, like what can you do to integrate more linear structures with cyclic structures? And I think that one thing, I mean, clearly what I was saying about the lead lag analysis, that doesn't require us actually to have an underlying circular structure. So I think one should be able to take that part of the story, because you're no longer worried about detecting circular structure, and then be able to say something about within a developmental process which gene is leading which other gene. I think that should be possible to extend to that class.
Cool, thank you.
You're welcome.
This is maybe a slightly different version of the first question, but hopefully it's different enough. I have, in a previous life, done some work on like nonlinear coupled oscillators and things like this. And so this last story here, where you're looking at the cell cycle and then also this regeneration cycle, it seemed like so at the outset of each one, you subset down to genes that you think were important. But if you were to, let's say, take the union of these gene sets, and presumably you do you believe that you would find, a second order loop, for example? And my question really is, when you have what you have here is a highly nonlinear relationship between the cell cycle and the regeneration cycle, where one is actually gating the other.
Yeah.
And you have genes that are probably going to be in common between the two of them. And I'm wondering if you have any thoughts on how you think the phase estimation would go if you had sort of two linked cycles that have a nonlinear relationship rather than the linear relationship which you had when you were doing your validation, which is easy because it's linear and it's all sort of sorts itself out.
Absolutely, absolutely. So I think there's potential to work these things out. I think the math is not going to be easy, but I think that it's worth doing. I think that. I mean, even moving beyond, thinking about, all right, the first Fourier phase is dominant and being able to ignore the rest. I think that's a little bit too naive, and being able to say, slowly build in more of these complexities. Now, and this is something that Kelly and I definitely agree on, that the really important thing, what's going to be really interesting, and this is a long-term project, is to think about interwoven phasing, interwoven processes, and understanding really how one impacts the other in terms of phase estimations and so on. It's not going to be trivial. It's going to be really interesting, but it's not going to be trivial. But because it's a really good question. And I don't know the answer yet.
Thanks as well for a great talk.
Thank you.
So you had some really nice visualizations, and so I have them in my mind. And so my question is, I guess you're in 5D, I can't do that, but let's say 3D. If I had 3D data that's going along like a corkscrew on a tube, what would your method say about that? And then if that instead kind of collapsed kind of like into a cone and they went to a point, what would your methods, would they find cycles sometimes? What would we see?
So I think that if it would actually, go down to a point, probably we wouldn't get a significant cyclic structure. But because it's really, it's an interesting question. Because the topology would sort of start seeing this as a point. But I think that if it's a corkscrew that doesn't actually converge to a point, I think that you would definitely pick up a cyclic structure.
Right, because you're looking for this S1 parameterization.
Yeah, exactly.
So as long as you have that.
Exactly.
And I guess, is that like an average across the depth of the hole? And so whatever.
Yeah, I think that's a good question. I'd have to think more about that, but I would think so, yeah.
Can I ask another question?
Sure.
Okay, so you simulated, in your first example, you had a simulated like ODE,
Yeah.
and then you tried to recover some structure about it.
Yeah.
Have you thought about, and a lot of times, I wouldn't be surprised if that ODE's parameters are pretty hard to recover from the data you generate. There are ways to do it, but I wouldn't be surprised if it's hard.
Yeah.
I'm curious, A, if you've tried that, but also B, after you do this analysis, do you think it tells you what kinds of generating dynamical systems or what kinds of properties those things have? In other words, like if we're trying to do system ID, we'd love to have a generating model that looks like physics.
Yeah.
Could these kinds of approaches tell us which models we should be designing and things like that, or properties of them?
So to the second question, I think that there's potential there for sure. I think that could be really helpful, provide an interesting perspective. Maybe not everything, but it could provide some interesting information. And then recovering all the parameters, I don't know. Because, at least it seems we can recover, as I said, the amplitudes and the phase shifts. Recover everything, I'm not sure. But at least that part of the story.
What is known about how PCA interacts with persistent cohomology? Because at the beginning you're going down to, okay, one is gene set analysis, and then but then you're doing PCA. So how robust is this?
So that's an excellent question. I don't know the answer. So I think that for the time being, in terms of robustness, we've seeing actually the sort of same cyclic structure showing up in different data sets seems to indicate that, okay, what we're doing is not unreasonable. Now, as a mathematician, I would love to have a proof that it actually is not unreasonable. I don't know, maybe somebody like Jose Pereira might know the extent to which this is stable under doing these sort of PCA projections. I don't, but real life experience seems to show that it's not a bad idea. I've learned something about being an engineer since I started doing this.
Okay, other questions? Oh, great. So thank you so much again for a
Thank you
wonderful
very much.
talk. Yeah.